This is episode 279 of the Stem Cell Podcast, Direct Reprogramming with Dr. Samantha Morris. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, on this little in-between episode, we got a special guest, Dr. Samantha Morris. She's on the show today to talk about her research on cell lineage and gene regulatory networks. We've also got our usual roundup of recent highlights in stem cell news. That's coming right up.
But first, do you work with human pluripotent stem cells? Get a free wall chart on the derivation and applications of HPSCs. To learn more, visit stemcell.com/HPSC-applications-wall chart. We'll start things off with a Nature paper. First author here is Nusaya Huda Gopi. This is coming from the lab of Muslifa Hanifa, but also some familiar faces on this particular paper. Sarah Teichman and Carl Kohler, who we've had on the show in the past. And if we're talking about Carl Kohler's work, you know, this is going to have something to do with the skin and the skin hair follicles as well, because that's what he's doing, making these hair follicle organoids, kind of these hairy organoids that we were chatting about. This is a prenatal skin atlas, which reveals immune regulation of human skin morphogenesis. So it's talking about how the innate immune populations, including the macrophages, are having a role not in their immune functions, but in the capacity and the capability of them to regulate skin development in vivo in humans. And so we know that the prenatal skin is populated by these macrophages, but that's what they're figuring out here. What's their role in morphogenesis? Okay. So here they put together a comprehensive multi-omics reference atlas of prenatal human skin. This is very early, 7 to 17 weeks post-conception, and combined all the best single cell and omics techniques that are occurring and happening right now, of course, like spatial transcriptomics, single cell RNA-seq, ultimately to characterize the tissue niches that are found in this very early skin. You know, again, 7 to 17 weeks post-conception. That's exceptionally early. And this is an atlas paper, right? So this atlas is ultimately showing that there's crosstalk that's happening between non-immune and immune cells, which actually underpins the formation of the hair follicles and is also implicated in scarless wound healing and critical for angiogenesis in the skin too. So I guess there's two sides of these atlas stories, right? One is just the atlas part and just seeking the crap out of whatever tissue you're interested in. But the interesting segment and the interesting application of these atlas stories is what you do next, right? And how do you can uncover different mechanisms of, like, for example, in this situation, skin morphogenesis. And that's what they're doing here, figuring out that there is indeed this crosstalk between the non-immune and the immune cells to determine the formation of the hair follicles. And then, you know, I think what really makes this story exciting is the the quote Carl Kohler element, right? You bring in this hair-bearing skin organoid, what they call SKOs, which are created from human embryonic stem cells and iPSCs, and they transform those organoids into, of course, the organoids that have these hair-bearing skin. Compare them to their models, to compare it to the data set that they got from this 7 to 17 week post-conception skin. What they found is that the SKO model actually recapitulated the in vivo skin epidermal and dermal cell types during hair follicle development, expression of genes implicated in the pathogenesis of genetic hair and skin disorders. So that's great. It's a good model. That's what we want from our organoids, to be good models, but it's not a perfect model. No model is perfect, right? So this SKO hair-bearing skin organoid model lacked specifically certain immune cells, and actually because of that had markedly reduced endothelial heterogeneity and quantity. So it looks like because of the lack of those immune cells, there's not as great angiogenesis, I guess you could say, or endothelial capabilities in those organoids, preventing them from going to the next step, which kind of makes sense. Ultimately, this in vivo prenatal skin atlas is showing that those immune populations, the macrophage derived growth factors associated with those macrophages, actually have a really important role in driving endothelial development and angiogenesis. So, this idea of vascular network remodeling was enhanced following the transfer of these autologous macrophages derived from iPSCs into the SKO culture. So they had the iPS derived macrophages, stuck them into the SKOs, and actually showed that you had improved vascular remodeling. That's awesome. That's really cool. And I think it's gonna probably take Carl Kohler's model organoid hair organoid systems to the next level. This is, I think, a really great paper because it's showing that these innate immune cells have a function beyond just their original assumed function in the immune system, but actually play a key role in skin morphogenesis, function that they achieve through crosstalk with the non-immune cells that are responsible for developing the skin in vivo. A lot of different elements to this story, cool model systems, the hardcore omics atlas, but I think one of the best things about this is how you're uncovering some really important mechanistic details about how immune populations and non-immune populations are interacting with each other in the context of human development.
Yeah, this is some next level type stuff here. A really great story, and I think a reflection of in this show how we're talking about in terms of themes. I guess some underappreciated or occult or overlooked roles of accessory cells that you wouldn't have guessed, and not just in early the incipient development, the first stages and induction of cell types in their initial spatiotemporal organization. But here, I like about this story that this idea, and conceptually, I think it's important to recognize in our field who focused on these, cellular primordia that we're trying to make into functional organs, that the developmental process is not just, gastrulation and organogenesis, but there's all these complementary roles of, in this case, immune and other cells too. A lot of immune, though, a lot of these immune cells coming through and instructing during these intermediate stages to direct them toward a more mature functional state. And I love the way they came around here to these skin bearing organoids. I mean, I remember, Arun, when I first met you, it was on the same day when Carl Kohler in California dropped his skin-bearing organoids in the ISSCR meeting, and people were literally chasing him down the street, just trying to have a conversation about it. So it's really gratifying for me to see how this model has, it was a big splash because it was like, wow, look at this meta element that's familiar in terms of the hair, and for all those male pattern people out there, a little ray of hope. But like, look at this like next level function here, and then coming back and using it as a tool to not only show, and you know, I think very clever and fortuitous maybe a bit, but to show using the fetal seek, as you said, they seek the hell at that fetal stage, showed what was missing in the organoid context, and then rescued it. I mean, wow, so cool. And I think there's a lot to be learned just generally in terms of organoids. And this isn't a new idea. I think a lot of people are going in this direction. But yeah, the role of these accessory cell types, essentially, circulating cell types on the progression of maturation of organ function across the board and all organ compartments, I think, is a really emerging and important idea.
Yeah, this is a complex story with a ton of different elements. And, it's also reflected on just the number of authors on this story. It's, as with any Atlas story, there's quite a few authors here, but you got folks ranging from great organoid biologists like Carl Kohler. You've got Muslifa Hanifa. Also, you have Barbara Troutline on this paper, who has got tremendous expertise on everything computational analysis and was, I think, one of the recent recipients of one of the ISSCR awards not too long ago. Really deep mechanistic dive. I actually didn't even remember that that the day we met at that ISSCR 2019 in Los Angeles, that's when Carl Kohler took the world by storm and showed those hairy organoids, the skin bearing organoids. And it's great to see his development as a scientist and his contribution to the skin development field as well. No doubt that people are going to continue chasing him down the hallways at the next ICCR, I'm sure.
Yeah, 100%. If I see you in the street, Carl, I'm going to chase you down. So I hope you brought your running shoes. Um, Next, I alluded to this. I'm going to a story. This is cell stem cell story. Same idea, similar idea, but kind of other side of the pole here, talking about pancreatic development. Um, it's well described. All organs, a lot of cell types contribute to the niche, the developmental niche. And in the pancreas specifically, it's been shown that there's crosstalk between the epithelium, mesenchymal, vascular cell populations. But and this is why this is a theme of the show. There's been little attention paid to the role of immune cells in the support of pancreatic organogenesis. But they know that they're probably doing something, right? Immune cells, they migrate and colonize the fetal organs across the board to establish peripheral tolerance. It's a well-defined mechanism of establishing that tolerance. And that function is impaired in a lot of autoimmune disease, namely type 1 diabetes, huh? So, apropos to the story that we're telling right here. But there's also evidence suggesting that immune cells, macrophages specifically, actually are instructive during organogenesis. So if you look at like the very first wave, yolk sac hematopoiesis, those erythromyeloid progenitors, they give rise to macrophages that colonize the embryo. And they colonize all the developing organs, as I just mentioned. And that's like the founder population of these so-called tissue resident macrophages. And in the pancreas, in the mouse, it's been shown that if you deplete these colony stimulating factor 1, CSF1 dependent macrophages in these mouse knockouts, you see that it actually undermines the morphology and the cellular composition of islets, of the pancreatic islet development. Also, if you take these CCR2 positive macrophages and do these kind of co-culture experiments and depletion experiments, it's suggested that they're necessary for islet proliferation and maturation perinatally, right? So a lot of evidence to suggest that immune cells, specifically macrophages, may have a role in pancreatic development. And in this story, as I said, a cell stem cell story from the lab of Maria Cristina Nostro, who's at the McEwen Stem Cell Institute in Toronto. They seeked the hell out of it. I mean, it wasn't a resource, it wasn't an atlas, but they went deep on the seek anyway, with a lot of single nucleus RNA-seq in this case of second trimester human pancreas. And they found there's a lot of hematopoietic cells and two distinct subtypes of these tissue resident macrophages. And then, I mean, when I say they seek the hell, they went super deep, more so even than your fetal skin atlas there. Literally 6 figures, maybe 5, a lot, almost all the figures in this paper were just seek after seek after, just looking at the macrophage, looking at the pancreas, looking at different stages of development and the secretome of these macrophages. And then they differentiate, they generate these endocrine macrophage organoids, as they call them, from human embryonic stem cells. And they show that these macrophages can actually support, improve, augment the differentiation viability of endocrine cells in vitro, and also increase enhance their engraftment potential. So, I mean, a role for immune cells in the pancreas. I mean, I don't think this is as comprehensive an analysis as you just described. Namely, there was not a lot of qualification of those ES-derived macrophages on the back end to see what their kind of phenotypic proximity to the native fetal macrophages are. But regardless, whether or not this is like the real macrophage and the real islet, and recapitulating the model perfectly, as you said, all models are wrong, Arun. I think the important advance here, which we kind of already knew, but I think it's important to put your finger on it from like a conceptual standpoint, is yes, that there is a paracrine role for these circulating, in this case, macrophage immune cells in the instruction and progression of organogenesis and tissue maturation. But also from a practical standpoint, it's like, yo, let's get these things into the diffs. I mean, we've come a long way with pancreatic islet differentiation. I don't think anybody needs a ton of help there. But in terms of like analyzing these specific islets, differentiating these contexts in terms of their fidelity to a physiological correlate, either fetal or adult, I think are some really important and interesting questions that may come from this story.
Yeah, it's a story that's following in the same vein of the one that I just presented. You have the role of basically an auxiliary cell type like a macrophage and contributing to development. Really exciting stuff here. I mean, I think one thing they alluded to in the limitations is sort of obvious, where they're limited by the sample size of the developmental stage they analyze. These are really tough data sets to obtain, which is, of course, the case with any sort of developmental atlas. It's so precious to obtain these samples. And here they're just restricted to the first few weeks of fetal pancreatic development. Ideally, you would want to go way beyond that. Some limitations with the single cell RNA-seq or single nuclear RNA-seq, which is, of course, only detecting the really highly abundant RNA transcripts. And there's not a ton of spatial transcriptomic data here as well. I mean, spatial is the hot, cool thing these days. And undoubtedly, they're going to incorporate that into the next iteration of this. But still, regardless of those limitations, just a really powerful model system. And once again, reflecting on this idea of how these auxiliary cell types, like macrophages, immune cells, can contribute to proper development in vivo.
Yeah, I mean, it's a good point about the the single cell seq in terms of limitations there. And what jumped out at me also here is, there's been some there's it's hard to get these tissues, but there have been uh studies that looked with single cell RNA-seq at fetal pancreas. So I wonder, like, if you go back into those data sets, as you said, like the phenotype transcriptomically speaking, from the single nucleus RNA-seq is going to be less robust, right? They're going to be a lot of transcripts that really just completely fall off. If you've done the single cell versus single nuclear, like you'll see transcripts that just disappear because of where they are, compartmentalized in the cell. So I wonder if you went back to those SC RNA-seq instead of single nucleus, would you find these same macrophage populations? And I think it speaks to the enormous potential of this era of high resolution transcriptomic and other epigenomic, all the omics, because these data sets are evergreen. And I mean, we're living in I think, an era where you can have almost instant validation from, at least from a like in silico level, at least, of your ideas and your observations, but also a platform for discovery across the board. So I for one, I never saw a single cell RNA-seq data set that I didn't love. So I might just mine these old data sets and see if I can get into these macrophages and see if I can validate it. Not doubting you, not doubting you over there. Just curious, Dr. Nostro.
Well, I, for one, have seen some single cell RNA-seq datasets that I do not love. It's very much dependent on the data sets that you're utilizing in the context of these grand compilation studies. I like what you're getting to, because I think we're at this point that we have so many of these atlas stories out there. We have such a massive repository of single cell and just in general omics data sets that we are, of course, incorporating our new best friend and everybody's new best friend, Mr. Artificial Intelligence, into really
Mm.
diving deep into all these data sets, compiling all of them, and figuring out what are the unifiers here, because this is way beyond the scope of what any single or even group of bioinformatics folks can do. This is something that I think, is perfectly positioned to do, is to incorporate all these data sets together and tease apart the connections, the mechanistic underpinnings behind development. And it's, there is, like I said, the limitation of the quality, right? Not
Mm.
every RNA-seq data set out there has high quality, But hopefully now we have enough data sets where we can perhaps compensate for that lack of quality in some of the earlier data sets, perhaps. So we gotta utilize the tools as they come out, and I think artificial intelligence is really perfectly positioned to intersect with these data sets and these technologies as well. So moving on to, I mean, this is a roundup focused on big scale, like high scale, high throughput, big data, sort of what modern science and modern biology is all about now, right? This is a paper coming from my guy, Joe Wu. And 10, 15 years ago, Joe Wu was not a big data guy. He was you know, let's make iPS cardiomyocytes the best we possibly can, and then use them for in vitro disease modeling with a sprinkling of drug screening built in there. Now, Joe's lab has certainly transformed, and he has become a leader in high throughput drug screening, incorporating some of these artificial intelligence approaches that I'm talking about. He actually has a startup company. Of course, as a Stanford professor, you got to have a startup, right? His startup is using iPSCs in combination with machine learning to uncover new drug targets. It's a Greenstone Biosciences up there in Northern California. And this is a paper where they've collaborated with that startup from his lab, in addition to the folks at the Stanford Cardiovascular Institute, to uncover a new therapeutic target for cardiac fibrosis. I mean, Joe is a cardiologist, he's a cardiovascular biologist. This is an area of study that he's been focusing on for a long time now. This is a Cell paper. First author is Hao Zhang. A bunch of folks who I actually don't recognize because I've moved on from the Joe Wu lab many, many moons ago. But Joe is doing still really great stuff. And like I said, the title of this paper is, multi-scale drug screening for cardiac fibrosis identifies MD2 as a therapeutic target. I like what they did here because they're incorporating all the strengths of the Joe Wu lab, which I'm very familiar with, of course, right? They incorporate the drug discovery, the iPS cardiomyocytes at the core, and incorporate this machine learning approach as well. So at the bottom line is, this is a story about cardiac fibrosis, and cardiac fibrosis is still in this year of 2024 a major concern when it comes to impairing cardiac function after, say, a myocardial infarction. You, of course, have scarring of the heart tissue after the heart attack, really impairs the function of the heart. But there's not a lot of effective clinical therapies for cardiac fibrosis and fibrosis in general. I mean, fibrosis is something that happens to all different types of tissues, right? So, to address this unmet need, they employed a high throughput screening system for antifibrotic compounds using iPS derived cardiac fibroblasts, which, funny enough, is a differentiation protocol that I actually started to work on during the tail end of my time in Joe Wu's lab as a grad student about 10 years ago, incorporated into one of our, cardiotoxicity studies. But here they've developed a fluorescent reporter ACT2 cardiac fibroblast line from iPSCs, which actually used in their high throughput screen. So they did a initial screen, finding some initial candidates using iPS cardiac fibroblasts, as well as cardiomyocytes and endothelial cells to figure out the specificity of some of these targets, specifically on the fibroblasts and not on the cardiomyocytes and endothelial cells. And importantly, excluding hits they found that were associated with cardiotoxicity. Because, of course, you want a drug that's doing what it's supposed to do on target, but is safe off target, right? You want it to affect the fibroblast specifically. So the screening process, which was done in collaboration with the high throughput screening facility at Stanford, which is an amazing, amazing facility, identified this compound called artesunate as a lead compound. And following pro-fibrotic stimuli, this artesunate actually inhibited the proliferation, migration, contraction in human primary cardiac fibroblasts, reduced collagen deposition, and improved contractile function in 3D engineered heart tissues. So using multiple model systems to show the same thing, that this drug is really effectively targeting specifically the cardiac fibroblasts. This artesunate also attenuated cardiac fibrosis and improved heart function in a heart failure mouse model. You got to have the mouse model if ultimately this is going to be a Cell paper. And then they dove into the mechanism, which of course is what you have to do. And they found that this artesunate specifically targets the myeloid differentiation factor, MD2, that I alluded to earlier, and inhibits TLR4, toll-like receptor 4 signaling, which ultimately alleviates the fibrotic gene expression in the cardiac fibroblasts. So ultimately, leveraging these multi-scale drug screening approaches, as well as actually some in silico simulations, which I didn't dive into a lot here in this recap, but that's where they collaborated with Greenstone Biosciences, the startup company, to do in silico simulations of how this drug is actually going to be impacting cardiac fibroblast function. And like what we talked about, that depends on good data sets. And so I'm sure the Wu Lab has a ton of cardiac fibroblast data sets to build on top of. Um, and ultimately, yeah, incorporating all these really cool approaches, iPSCs, tissue engineering, animal models, multiomics, to identify MD2 as a potential therapeutic target for cardiac fibrosis. This is a very Joe Wu Lab paper, just throwing a bunch of different model systems at the approach, but then taking a deep mechanistic dive as to how this artesunate is actually inhibiting and alleviating cardiac fibrosis, which is a problem that's universal. It's a big unmet medical need. So congrats to Joe and his team for another big time study.
I mean, this is great news for patients. It's not great news for anybody who's studying cardiac fibrosis and like therapeutic means of addressing it, because Wu Lab fixed that for us, and now we can all just move on. Joking aside, this is a really big deal, right? Although we say that a lot on the show, like, oh, this is a huge deal. And like, would I be surprised in 10 years if I never hear anything about artesunate and therapy again? I mean, no, probably not, because I will have forgot about it by then. But like from where I'm sitting right now, it seems like this is it, right? It seems like this is something that, I mean, could be in trials straight off. This art, as they call it, it's been in play for 50 years, treated millions of patients in treatment of malaria, which kind of raises a question to me: what's it doing? What's the common, the overlap there in treating malaria and cardiac fibrosis? But I might take malaria if it means I could get my hand on some of this art. And the other thing is these mouse models that you mentioned, like they were able to change the landscape in terms of fibrosis, that are therapeutic, even in mice for whom the fibrosis was already set, right? The dysfunction was already well established. So, I mean, that's new to me. I thought it's kind of like you can't reverse the scar, but hey, here we are. So I think this is a huge deal, and I'm interested to see what kind of moves he's making in terms of trials for this. Like, considering it's already been proven safe for malaria, does that mean like you can off label it? I mean, is that going to accelerate? Are we going straight to phase 3? I would love to see what this can do for people.
Yeah, I think you're exactly right. This is a repurposed drug study, right? So it's already gone through the rigors of clinical trials once, and of course, the early phase clinical trials are all about safety, efficacy, and this is a malaria drug which has been established for a long time. So that apparently those safety profiles are good. FDA loves that. They love repurposing something that they know is already safe. So yeah, I think moving it towards the later phase clinical trials is probably easier in this context. Um, and yeah, what one thing that you mentioned is important, right? This seems like it's working, but what happens 10 years down the road in the clinical trials? We don't know. But the way you kind of hedge your bets in a way is to have a bunch of model systems, right? You use all the model systems you possibly can to verify that this artesunate is doing its thing in alleviating cardiac fibrosis, inhibiting the function of cardiac fibroblasts. You got the in vitro, iPS derived, animal model, in vivo, in silico, all the in whatever, in vivo, vitro, silico, everything you can think of has validated that this works. And the only thing that's left to do is to put it in people. And inevitably, that's what they're going to be doing at Stanford.
Yep. In everything science, but started at least this study using a pluripotent stem cell platform. And that was the dream. And here we are. It is reality. So a really exciting application, been a long time coming. Congrats to the Woo Lab, and congrats to you too, Arun, for having such a fine pedigree. As I said earlier on, this show is about like who would have thought that I got that malaria and suddenly my heart is stronger, and all the other who would have thoughts here's the final chapter in the occult knowledge, and it's from Berna Sozin, who famously came out of Magdalena Zernicza Goet's lab. I learned that I've been pronouncing that incorrectly for many, many years. My apologies. Sam corrected me on that. You're going to hear from her in just a few minutes. But first, this final roundup story. So yeah, Berna Sozin, she came out of the Magdalena Zernicza. I screwed it up again. MZG lab about 3 years ago, started at Yale. And as soon as I mention her name, of course, you're waiting for me to come with some STEMbryo story, right? But that's not this. That's not this. It's an embryo base, and it's really basic and fundamental, super elegant, beautiful story. But it's refreshing an idea that actually came onto the scene like more than 100 years ago. And this was this idea of a like metabolic gradients in development. This was initially put forth by Charles Child, literally in 1915. They're still talking about protoplasm back then. He has this chapter in this book called Metabolic Gradients in Organisms. And then that kind of went away, right? We discovered morphogens and all the other stuff. And that whole metabolic overlay idea kind of went away. But now what's old is new, and metabolics and metabolism's back, specifically in terms of impact on development and organ homeostasis, etc. And here the question is how an organism establishes a positional identity, right? And of course, there was that gradient theory that I alluded to there from Dr. Childs, where there's this idea that a graded metabolism along the axis directed tissue patterning. And more recently, as I mentioned, this idea of metabolism has become, du jour again. These metabolic enzymes, also metabolites, in and of themselves, can function beyond their like bioenergetics, that nutritional role, to actively modulate or instruct developmental programs. And this has been shown in chick and mouse embryos that there's these regionalized glycolytic gradients, right? So there's evidence to support it, but there's still questions in terms of like in the mammalian embryo specifically, whether nutrients are processed in like a uniform way or a random way, or if metabolic signaling might be linked to morphogen signaling. And if so, how are these two inputs integrated during early post-implantation morphogenesis? So it's tough, obviously, to do this in embryos, to do both, right? To look at both the metabolic as well as morphogen signaling kinetics at high resolution in a live embryo, developing embryo. Hence, you thought I'd come in with the STEMbryos, right? And of course, that is an application I think is going to move from this to get it into actually a human embryo model system. But here, they're looking at this the instructive role of cellular metabolic activity in mouse gastrulation in developing embryos, also looking at these stem cell as well as embryo-derived tissue explants, right? And what they show is that there's these two distinct, spatially resolved and stage-specific waves of glucose metabolism during gastrulation. And they do this with really beautiful images. I mean, it's just a real treat just to look at the imaging in this paper. I would encourage everyone to have a look. It's a Nature paper, just came out. And interestingly, this was submitted like a week after last year's meeting in Boston, like when there was a big splash about these human embryo models and everyone was going crazy about it. That's when this paper was submitted. Just came out last week. Yeah, and they show that there's this first wave of glucose metabolism that occurs through this hexosamine biosynthetic pathway, and that's critical for driving fat acquisition in the epiblast. And then there's a second wave that uses glycolysis during mesoderm migration and lateral expansion. So, really cool just at the outset to make that link between metabolism and morphogenesis, but also they then connect mechanistically this metabolism and glycolysis to ERK activity in both the first and second wave, um, and show that there's like a synergy between the morphogenic gradients and the metabolic. And I think really provides strong support for this idea more than 100 years old, that metabolism is critical to guiding cell fate, and also to endowing specialized function during development, which I think is a great, it's always great for me when you think one thing is doing one thing exclusively, and it's like, oh yeah, by the way, we're also doing that. And like, is there anything more, monolithic than nutrition and metabolism? Not so much anymore. I know we realize that there's all these ancillary roles of metabolism and instructive roles, but here, like at the real root, at the very beginning, in the earliest points in cellular specialization, spatiotemporal organization, the embryo, metabolism is playing a critical role. So a real important conceptual event, one in a series from the Sozin lab, who came on the scene, like a rocket, and continues her ascent.
Really exciting work here, kind of building on the theme here on the show. One of the themes, I suppose, of this roundup is uncovering the early mechanisms of early development using cutting edge tools. This is I think, a really important story, and the first thing I thought of was not directly related to development per se, but perhaps how some of these findings could be applied to situations where, quote, development goes awry. And I'm thinking of cancer, because cancer is kind of the opposite of development, and things are going awry in that situation. Of course, as we know, metabolism and cell fate decisions in cancer are highly influenced by metabolic states. So I'm wondering if some of the findings from this story can be applied in that context as well, but also even in other situations of quote development or analogs of development, like regeneration. What's the role of some of these metabolic pathways and glycolysis metabolism in regenerative processes as well? I think there's a million different ways you can take this foundational study and bringing it back to the tools. And one thing they allude to here is, I think, one technology that would really facilitate the next wave and the next iteration of this study is single cell spatial metabolomics,
Hmm.
right? Spatial metabolomics, really figuring out what's the metabolic profile within individual segments of tissues. I think that's going to take this study to the next level.
Oh yeah, and also, doing this in the I'm not going to say stembryo. I'm like, now I'm saying, I was saying it, mocking it. Now I'm just fine. I'm saying it. So I'm not going to say it. In human, also the mouse embryo models, I think it's going to be a really great counterpoint to this, because I know a lot of this stuff was doing the live imaging in vitro and all that stuff, but the embryo models are totally different, right? Devoid of any kind of like the extra embryonic tissue and the investment of the maternal interface, all that stuff, which may have an impact on the metabolism and nutritional support, all that. So like I'd be interested to see how these axes are defined in the human embryo, mouse embryo models. And if they're not, is it cell intrinsic, so to speak? Are these waves that were described, do they come from within the embryo, or is there extrinsic cues that direct it? And if it's the latter, then I think it's going to be really critical for us to move these embryo models forward to try and understand how that metabolic kind of spatiotemporal axes are situated in the model, and whether or not they're faithful to the real thing, if we're really trying to approximate human namely embryogenesis with high fidelity. So a really interesting follow-up that I'm sure is right now in the process of being done. All right. Well, we talked about fate like for the last 30 minutes. Now we're going to talk about fate some more with Dr. Morris, although a different spin, and I think a really important spin that she brought to the fore years ago and has built on since. Can't wait for that conversation. Before we get there, though, I got a quick message from STEMCELL Technologies. Take your human pluripotent stem cell cultures further with M-Teaser Plus from STEMCELL Technologies, the most widely published medium for feeder-free human ES and iPS cell maintenance, is now formulated for enhanced performance and versatility. M-Teaser Plus reduces medium acidosis for more stable cultures all weekend long. To learn more, visit www.stemcell.com/mteaserplus. All right, you guys, with us today, we have a scientist on the move, Dr. Samantha Morris, who is currently, I guess, still at Washington University in St. Louis, where she is associate professor of genetics and developmental biology. But the lab is in the midst of moving to Brigham and Women's Hospital and Harvard Medical School, the Department of Systems Biology. Research in the Morris Lab focuses on dissecting and manipulating gene regulatory networks that define cell identity and applying this knowledge to engineer clinically relevant cell types. They also develop new single-cell technologies to map cell lineage and identity in parallel to better understand how cell identity can be reprogrammed. Dr. Morris, thank you so much for joining us on the show today.
Thank you. Great to be here.
Yeah, thanks for being here. I mean, we've heard so much about your work over the years. It's finally great to have you on the podcast. Like Dalen mentioned, your lab uses all these really cool new technologies and tools and single cell to dissect the gene regulatory networks, determining cell fate. And before we dive deeper into the weeds of, the papers and all that, why don't you get us started by giving our listeners like a broad overview of what your lab does, kind of in your own words, and maybe kind of how you got here too.
Okay, so I'll try to wrap that up with a bit of backstory as well. I am a developmental biologist by training, so I got into this by way of embryology and understanding how cell fates were specified, specifically in the mouse embryo. And I became very interested, so back when I was at the University of Cambridge, in how we could force changes in cell identity. And this was around about the time of the Yamanaka factors being published, and just realizing that we could force these changes in cell identity for eventual therapeutic purposes. A lot of excitement in the field at that point. And at that point, I moved over to Boston for the first time and joined George Daly's lab and started a project on cell fate engineering, and realizing that switching one identity to another, specifically via transcription factor overexpression, it's not as simple as advertised. And I've been building my lab now for the past almost 10 years on this problem. How do we effectively engineer cell identity so we can faithfully recapitulate those target cell types? And I think it's such a challenge that it's forced us to go back to the basics of understanding what cell identity is itself and how it's defined. So almost taking me back to my developmental biology origins. And to tackle these questions, it's taken us into the arena of technology development, which I absolutely love. So single cell experimental genomic technologies, computational technologies, and understanding fundamentally how cell identity is wired.
Yeah, I appreciate the brief bio there, because reading into your story, I was really captivated by that arc. I mean, I love my own life in science. I love my life, period. It's a great life. But if I had to rewrite my scientific narrative for a made-for-TV Netflix type movie, because I don't know that anyone would tune in for as great as it is for me, I don't know that anyone's tuning in for that. But if I had to rewrite it to spice it up a bit, it wouldn't be far off from your own. because it's familiar also to me. I mean, you did your PhD at Cambridge in the, post-Gurdon era. Gurdon was way back, but the era in which he was appreciated for, as you alluded to, the Yamanaka factors. So the enthusiasm around Cambridge and his work and developmental biology and stem cells was there, similar to my own experience growing up with stem cells when they were first, introduced to the world, human at least. And then a postdoc with Magdalena Zernicka-Goetz, the notorious MZG, whose imprint I think nowadays is much larger perhaps than it was then. But I mean, those ideas were floating then. So you were there really at the inception, at like the nucleating stage of all those amazing ideas that are currently, culminating in all these amazing stories. And then, as you said, with George Daly, so a great arc working with early days chick, xenopus embryos to modeling human development nowadays as like a series of cellular systems is how I appreciate your work. In that arc, it's a really fine illustration of the progress the field has made at large.
Mm-hmm. And I love that you mentioned John Gurdon there. He was a huge influence back when I was in Magda's lab. I was fascinated by his work in the 50s. And, really, think of it as, bookending the Yamanaka factors, with those early examples of how we can change cell identity. And I remember one of the hallmarks of the Gurdon was we'd have tea time. And I think one of the hallmarks of British science that I certainly miss in the US, we'd have tea in the morning, we'd have tea in the afternoon, we'd have lunch, we'd go out for a beer after we finish experiments for the day, which was fantastic. But I'd often sit down with John and bounce my ideas off of him. And one day he said to me, I think you're really interested in reprogramming, Samantha. And we got into that because I say to him, I want to change this cell type into this cell type in the embryo to really understand how these fates are specified. And he said, I think you're interested in reprogramming. So I bounced this idea off of him. I said, well, maybe I should move to Boston. It seems like an epicenter of reprogramming right now. And he agreed. So he was encouraging me to move to the US at that point. So I love that you bring up John in this conversation.
Well, we've had him on the show, and I mean, he's at once this icon, but also a really benevolent, avuncular influence, and so we love him here, and we're not alone. But there, I mean, you said it. You went from training with and at the knee of so to speak, but really direct one-on-one mentorship with the prime architects to now. I mean, I think it's fair to say that you're amongst the architects of the field, however humble you want to be about it. So what would you say in that arc, which is still, I would, on the upward trajectory? What's one thing that you've been really surprised at so far in the progression of the field, or any aspect of the field?
One thing that surprised me about the field came about in, I'd say, 2013, 2012, when I started work on CellNet with Patrick Kahan in George Daly's lab, and this was in collaboration with Jim Collins' lab. And as I said, there was incredible excitement around reprogramming and the potential of this. And we built CellNet to test the quality and the claims that people were making around these reprogramming protocols. A flurry of high-impact papers claiming, oh, we've made hepatocytes, we've made neurons, we've made cardiomyocytes. And with CellNet, we put that to the test, and we found that very few protocols were actually making what they were claiming. And these claims were based on a handful of cell type markers, and really no in-depth, systematic and unbiased way to measure cell identity. And that's what we achieved with CellNet. And it surprised me over the past, coming up to 15 years now, that people keep maintaining these claims that they've made protocols that can specify certain cell identities with high fidelity, high efficiency. And that's really not the case. Or they're selling approaches that can get you to your target cell identity. And, these approaches really need a lot more work. And as I mentioned, the arc of the lab is, this journey is taking us back to the fundamentals of cell identity and what it means to be a certain cell type. I do not think it's a trivial problem. So it continually surprises me that there's a gulf between what people claim and what is actually feasible, and that I think we need to take a step back and ask, how do we truly get there for cell engineering purposes?
Yeah, I think what you're doing is so critically important. And I'm not calling you like the reprogramming police or anything like that, but I think having that sort of validation is so critical in our field. As you know, stem cell biology recently and over the last couple of decades has had a bit of a black eye because of the things that you've described, right? I mean, I'm a cardiac biologist, and for a long time, everybody thought you could turn mesenchymal stem cells into cardiomyocytes and use them to improve cardiac function. And we knew that was, of course, not the case, right? So having that proper validation of what one cell type is turning into is so critically important. And you talked about some of the methods like CellNet that you use to flesh this out. But fast forwarding a little bit more recently, you actually had a recent paper in Nature Biotech, which I think we actually covered here on the show, where you developed a new approach called cell tag, which is enabling direct capture of different barcodes expressed as polyadenylated transcripts in both single cell RNA sequencing and single cell ATAC. Again, sort of in the same realm of what we're talking about, looking at cell fate, but more of, looking at independent clinical tracking, transcriptomic, epigenomic cell states, really being able to do lineage tracing at a depth that hasn't really been done before. And like what we're talking about, you importantly demonstrated its validity for tracking cell fate during hematopoiesis, as well as in direct reprogramming of fibroblasts to endoderm progenitors. So that validation is really, really critical. Tell us a little bit more about this new system, this cell tag system, and maybe what sort of fate mapping you hope to apply it to in the future.
Yes, back when I launched the lab almost 10 years ago, that's when I really got into the high throughput single cell profiling methods. And at that point, we were working with DropSeq. And I say we at that point, it was just me in the lab. So before I'd been able to expand and employ people and convince them to join my group, I got DropSeq up and running and just playing around with that data and looking at barcodes for hours on end. I had this idea, well, what if we put a random barcode into these cells using lentivirus, and that would allow us to track these cells over time. This is a very active area of research. Lineage tracing as a field goes way back, and fascinating field. There's a lot of groups, developing these lineage barcoding methods. But the cell tag approach is extremely simple. So back then, using this very short random barcode, using lentivirus to introduce it into cells. And the reason I was so excited about this approach was because, particularly with reprogramming in an accessible in vitro system, we could tag the cells when they were fibroblasts, then plate the cells, let them grow for a while, so a few cell divisions, then hit the cells with the reprogramming factors, so with the IEP reprogramming, FOXA1, HNF4 alpha, then allow the cells to reprogram, and then we would introduce a couple more rounds of barcoding so we could build rudimentary lineage trees. And at the end of the reprogramming process, capture the cells and then be able to track these lineage relationships back through time. We could start to understand what are the early changes in these cells that set them on a specific trajectory. And back with the original cell tagging paper that was published in 2018, we found these two distinct trajectories. So one to the reprogramming fate that we've shown is responsible for functional engraftment of the intestine. And there's also this fate that we call the dead end. And the cells go into a mesenchymal-like state. They don't fully epithelialize. It's a reprogrammed state, or it's an off-target reprogrammed state. And through cell tagging, and particularly the cell tag multi paper that you mentioned that was published earlier this year in Nature Biotech. And we were able to capture lineage and parallel transcriptome chromatin accessibility. And this allowed us to understand those gene regulatory changes very early in the reprogramming process that assign cells either to this reprogrammed or dead-end trajectory. And that information allowed us to shift cells from that dead-end trajectory back onto a reprogrammed trajectory, so increasing the yield of cells that would functionally engraft the intestine, and that was shown in another study. So the cell tag project has been extremely fun. We continue to work on lineage barcoding methods now. New versions of the cell tagging approach are bubbling away in the lab, and hopefully we've got some preprints coming out on that next year. And it's just such a fun field to be involved in, the lineage tracing field in general, and many creative approaches now. Fundamentally, it allows us to link those early changes in cell identity to eventual outcome. As reprogrammers, that's extremely valuable for manipulating the process, refining it, increasing its fidelity, its efficiency. Therefore, we will always go back to these lineage tracing approaches. And I think it's a great example of how we are integrating technology development into the science. At that point, there was no technology available to be able to enable these insights into reprogramming. So we set out to develop that technology.
Yeah, I love about your work that, I mean, you said lineage tracing, it's expanding, right? But lineage tracing is as old as developmental biology. And what I love about your work, and it's just science in general, is that you recursively apply these, increasingly sophisticated tools that are the innovation in a kind of an old framework to bring finer resolution to like the oldest questions. In this case, talking about the lineage tracing, conceptually, I want to like bring that back to CellNet. Before the cell tag, there was a CellNet, which I would argue there's an obvious conceptual link there. And as you described, this was a story you published. It was like just about a decade ago now that it came out. And one of the pivotal conceptual advances there, for me, my takeaway, I remember when it came out because it was this idea that reprogramming through directly to the terminal fate, which I always thought was a little bit like too good to be true, just intuitively, versus going through this, progenitor intermediate and providing like clear evidence that going through the progenitor, going through this kind of, I guess, pseudo-natural process, you arrive at a more faithful fate. And of course, I think that the approach also has practical advantages in terms of getting a progenitor that can scale up, be proliferative, like transit amplifying state. And also the element of like the in situ responsiveness to the niche-derived cues. So like for me, it's like I don't want to call it a silver bullet, but it addresses a lot of the challenges, I think, to cell therapy and engraftment and function. But you had a nice synthesis there of how the direct reprogramming boom was not exactly, it wasn't. I would say it was more apparent than real, right? The actual clinical application of these directly programmed cells faces a lot of challenges, and as you just alluded to there as well, the off-target fates, right? So, where are we with that? Seems like a long time ago. We were talking about like direct reprogramming in situ of cardiac fibroblasts, that seminal Deepak Srivastava story, but like I feel like that kind of went away. Is there anything to that still? Have we moved on completely? What's the status of this idea of direct reprogramming in situ or in vitro and transplant? What's your take on the whole thing in terms of viability, feasibility?
Yeah, I love this question. Back with the CellNet analysis 10 years ago, that was one of the most faithful reprogramming protocols because the cells were delivered directly to the niche. The niche factors could then finish this partial reprogramming job. And this question leads us into another branch, another journey in the lab, following up on the original CellNet paper. And one of the main conclusions from that paper was that direct reprogramming isn't actually direct, it led to a hypothesis that cells do regress to some kind of developmental progenitor, and then we would have to differentiate them out from that state. So they certainly don't go back to pluripotency, but they do take a step back. This started a long project in the lab, and it ended up with 2 pieces of software. One's called Capybara, and one is called Cell Oracle. And Capybara is a cell type classifier. And we built the cell type classifier to work with or treat cell identity on a continuum. And most cell type classifiers work with cell identity as a discrete property, but we know as reprogrammers that this is a non-physiological process. Cell identities really go off-road in this process and aren't necessarily physiological. So we really were working with a smear of cell identity with Capybara, and it could capture these transitions and these multi-ID states that we observe frequently in reprogramming. But with Capybara, we take our query single cell profiles and we use existing single cell atlases as references. And with this hypothesis that during reprogramming cells regress back to a developmental state, we built this beautiful reference atlas of all mouse developmental single cell profiles that we could get our hands on. And we had no match of our induced endoderm progenitors. I mean, I called them endoderm or renamed them endoderm progenitors back in the CellNet paper thinking, oh, this is a developmental cell type. And we didn't find any hit. And then we considered another alternate hypothesis that these cells may be some kind of regenerative cell type and not exist in a developing organism. So we went to a reference atlas or damaged mouse liver, and we found that these cells more closely resemble injured biliary epithelial cells. And I think that brings up an interesting observation that these reprogrammed cell types might not relate to a developmental progenitor. They might be some kind of regenerative cell type. And this is something we are still exploring. It also opens up some interesting avenues in terms of cell potential. So we might be creating these cell types that don't exist in a normal healthy organism, but they have much broader potential than we expected. So that's one direction that we're going in. So back to this your original question of the state of in vivo or in situ reprogramming now. I think this is a really raises interesting questions about direct reprogramming itself. Because if cells are taking a step back or they're going into a regenerative state, then how are they behaving in this in situ reprogramming context? And I think with the early cardiac reprogramming work, we don't have any evidence to support this, but cells might take a step back into this developmental regenerative intermediate, but then the factors within the niche could fully differentiate those cells out to target cell identity. I think there's a lot of promise for in situ reprogramming still, but it's quite challenging. One of the reasons it's challenging, and I think we haven't seen much follow-up, is because direct reprogramming itself is such a rare event. So in most protocols, less than 10% efficiencies. Also in these protocols, we have these off-target reprogramming events, which we certainly don't want in an in situ protocol. And that's taking us back to trying to understand reprogramming origins themselves. And especially with all of these new technologies coming online, and we're developing the technologies to ask these questions as well. I am particularly excited about being able to dissect these successful reprogramming events and understanding what is it about that fibroblast, for example, that enables it to successfully reprogram. How do we trace back those early molecular events? And I think that has huge importance for in situ reprogramming as well. So how can we, increase the efficiency of that process and fidelity? I keep using these words a lot today, but efficiency and fidelity are absolutely key here.
Yeah, you're absolutely right. And, for obvious reasons, that Deepak Srivastava reprogramming paper is one of my favorites. Is actually one of the reasons I got into this field in the first place, because it's this kind of like a very unique approach at that time in 2010. where you go straight from one cell type to another. But as we're discussing here, it's perhaps not as straightforward as it might seem, not as clear as it might seem. But you're right. The efficiency of the process is really the number one thing that always stood out to me. And that's perhaps what's limiting it from becoming clinically translatable. I mean, the dream is you want to translate or transform the fibroblasts in a after post myocardium, post myocardial infarction on heart directly into cardiomyocytes, right? But we're very limited by the efficacy of that process right now. So moving on a little bit, we talked about all the cool technologies your lab is working on. You named a bunch of them that you've actually developed in-house. Capybara, which I love the name, by the way. Oracle, CellTag, CellNet. These are all things that you guys have been working on over the years. I mean, it's no secret that your lab is heavily computational, but maybe even perhaps the model lab for what the future of biomedical research is going to look like, right? It's a hybrid wet/dry lab, I guess you could say. And we've talked about the importance of reliable wet lab data sets, okay? Because you've described yourself as a traditionally trained developmental biologist, stem cell biologist who adopted and then learned these computational approaches, so you can understand and you can appreciate the importance of having really reliable wet lab data sets. I mean, it's something we talk about a lot here on the show with these computational models and especially machine learning driven approaches where if it's garbage in, it's garbage out, right? You got to have a good data set, good wet lab data set to really feed those downstream approaches. So I'm guessing you're not going fully in silico anytime soon. That's kind of what I'm getting at here. And there's always a need for talented cell biologists in your lab too, and I'm sure. But I'm a new PI, so I like to pick the brains of folks who have come before me in terms of deciding how do you pick the right people to join your lab. Like, what do you look for? Is having some sort of computational background a prerequisite? Do you try to keep things evenly split between wet and dry labs? So what's your secret? What's your approach to hiring new talent for your lab?
Yeah, this is a great question. And I get this question a lot from people applying to the lab, making an assumption that they need to be trained computational biologists to join the lab, which is not the case whatsoever. In the lab right now, we have an amazing mix of cell biologists and computational biologists. I refer to us as an amphibious lab. Everybody tends to do a little bit of both. Even if we have somebody coming in full computational, they eventually get sucked into the wet lab because there's always so much excitement going on in there. That's really where we test the hypotheses and generate the data for analysis. I like you saying that it doesn't look like we're going to go full in silico anytime soon. I completely agree with that. Cell Oracle is a really fun project to predict the outcomes of transcription factor perturbation in silico. But there are limitations to that approach. We can only predict one factor at a time. When we apply this to reprogramming, if we are reprogramming cells to a state that Cell Oracle has never seen before, we're not going to get any insights into that process. So there's still so much work to be done in this area. And this area of single cell perturbational omics is fascinating. I think it's an up and coming field. Particularly think about machine learning with combinatorial perturbations as well is essential for reprogramming. And maybe that's completely another discussion that we can get into later. But back to how I hire people in the lab. So to some extent, I let the lab's focus be driven by the talent that is coming to the lab. So if I see a great postdoc application, super talented person, very passionate about what they're doing, that's my primary focus. Does this person, love reprogramming? Are they interested in stem cell biology? Are they in a completely different field, but they're passionate about, our main goals in the lab? And then secondary to that, I will then look at their experience. Are they a cell biologist? Are they a computational biologist? And nowadays it tends to be a mix of both. But it's really driven by the talent of the people who are applying. And again, I think Cell Oracle is a great example of this. So Kenji Kamemoto, the postdoc at the time, so he's also just started his own lab at University of Osaka, so please check him out. Kenji came to my lab. I saw his application, he interviewed, and he's one of the most creative scientists I've had the pleasure of working with. Just absolutely fantastic scientist. And he's taken the lab in directions that I didn't, necessarily think we'd go into. And it's just working with people who are talented and have that passion, and trusting in them, and also our working relationship, and following those exciting avenues, that I think creates a lot of a lot of magic in the science as well, and in the lab. And I will continue this hiring strategy of just seeing what talent comes to us, what direction will it take us in. And so obviously there needs to be oversight in this. My primary job right now is to write the grants, bring the money in, write the papers, get the grants, and follow that cycle. So within that broad umbrella of reprogramming and the deliverables we have for the grants, we have to work within that. But beyond that, I just like to take our direction guided by the talent that's coming to us. And ask me again in 10 years how this is working. But for the past 10 years, it's been really satisfying.
Seems to be working out for you so far. And I mean, my judgment, and largely aligned with what you're saying, is that the best labs, in the best case, I would say, the best lab has a broad scope of technical and foundational knowledge that expands a postdoc's horizons and makes them a more complete scientist. And conversely, the best postdocs bring skill sets that then increase the scope of the lab's capability. And I think you're a great example of that, or your lab is a great example of that, because you are, as Arun described, at this kind of in between place, wet, dry, as you said, amphibious lab. I like that description. Although in your case, we're talking like not just Marines, but Navy SEALs at the level you're operating. And I think this is a key to the future of science, especially amongst the newer generation of trainees and PIs. And I think that, it's no coincidence that this is why you were named as a Robertson investigator by the NICEF. I was associated with the NICEF, the New York Stem Cell Foundation, as well as a Druckenmiller postdoctoral fellow about 100 years ago. So I'm familiar with that mechanism, and especially years ago when we were coming up, it really played a critical role. It was a much more hostile funding landscape for stem cell research, which has been, addressed in large part in more than a decade ago now with IPS technology. But nowadays there's human embryo models, and also in parallel, closely related though, there's increased political division around personhood, right? So this is like renewed this charged atmosphere, the similar atmosphere that necessitated the founding of like NICEF or like CERM, all these more independent funding bodies. It seems like we're kind of coming back around into that climate a bit. We actually talked a lot about this with Rowan Carvis and Thor Tunesson, who are your colleagues there
Next
at
door
WashU.
neighbors, yeah.
Well, see, there you go. So you know exactly what I'm talking about here. I'm working with human embryo models. This is a real, live wire for them. And of course, the taking special care because of how polarizing it is. But they shared that, St. Louis isn't exactly, while it's a permissive enclave there at WashU, more, wider, it's not exactly the most permissive for this type of research. So what's your experience along these lines? I know you're moving to Beantown. But just generally speaking, working at the point of the spear, so to speak, do you think that we're going to have to, or do you think we need funding bodies like the NICEF and CERM more than ever as support for this research? Are we treading into this more hostile or gray area territory again, where the independent funding bodies outside of the NIH are going to be critical?
Potentially, yes. And, I think we should always be prepared for that. And, another aspect of, these independent funders, so particularly NICEF in my experience, is they're there to support people rather than projects, in their words. So supporting the ideas of the PIs and just letting them be creative with their science. And I think that creativity, certainly can take you in different directions. So Thor's lab was neighbors, so we hear their concerns on a frequent basis. Our work is more mouse focused. We do a little bit of human work, so human IPS based, but not human ES based. But I hear Thor's concerns a lot in this. So I think it's incredibly important that we do have these independent funding sources, because the political climate can change at any moment. And we just have to be prepared for that.
Yeah, you're absolutely right. And, also, of course, the trainees benefit from having the secondary source of funding. I mean, I am a trained stem cell biologist because of CERN. And, I think it's a big reason why people come out here to the West to do stem cell biology, is because there's a secondary source of income or revenue or whatever you want to call it that's going to support this somewhat grayer area of study. I mean, there's folks out here who are working on, chimerism and all these different areas of interest that traditionally aren't funded by the NIH, right? So I think it's a really important conversation to have. Wrapping things up, before we let you go, we wanted to ask you, of course, about this big lab transition that you're in the midst of right now. I mean, we alluded to it a couple of times here on the show. You're actually heading back to your old stomping grounds in Boston, but also actually my old stomping grounds too. Actually, I was a postdoc at the Brigham and Women's Hospital at HMS. I was in the Seidman lab in the Department of Genetics. I think you'll fit right in. You're familiar with the area. There's some of the world's biggest names in genetics and systems biology out there. I mean, looking back, it was just so wild being just down the hallway from titans in genetics like George Church, Steve McCarroll, Olivier Porquier, all these amazing names that we, look at as icons, right? So no doubt that your colleagues there are going to be some of the best when it comes to systems bio and genetics. But, tell us more about that decision and what you're the most excited about to be back in Boston.
Yeah, I moved to Boston in 2011. So, a while ago now, and left Boston almost 10 years ago. I'm just excited to be back in the city and being back in that environment as well. It's a very fun, fast-paced scientific environment. I often reflect on, coming out to WashU, starting my lab here, being here, in those junior years. And I, really hand on heart could not think of a more wonderful place to start my independent scientific career. The environment at WashU has just been fantastic. We often say this, well, most institutions say this, we're very collaborative. That's really true of WashU. So it's almost like a little bubble. And to start your lab in that bubble, to have amazing mentorship and to be encouraged down a specific track has just been incredible for the growth of my lab. It allowed me to be more creative, I think, and a little bit more bold, because I felt so safe in this environment. And, forever be grateful to WashU for that. And now going back to Boston, I'm excited for this phase. So now that we've built our research program, we're ready to take it up another notch. And I think the team right now are just going to thrive in that environment. So particularly excited for that, and the new colleagues as well. Also old colleagues. There's so many people we know in Boston, and just excited to work directly with them now.
Again, going back to my made for Netflix movie, limited viewership for sure, but I like this point in the arc because yeah, WashU is such a great incubator, and they've done so good at identifying and cultivating young talent, and the evidence isn't all the productivity, but now it's time to take your place. Alongside all the titans, as Arun described them, but at a place which has really been, I would say, the nucleus of stem cell research. I mean, there's the site Wisconsin, of course, and the West Coast. Arun, you get some credit to a lot of good people over there, but Boston is really, I would argue, the nucleus of stem cell research over the last decades. And you're going to be a great fit there. So congratulations on the move.
Thank you.
And before we finally let you go, we have a couple last questions, science peripheral for our listeners. First is, if you were not a scientist, what would you be doing with your life?
Yeah, I've always, from the beginning, thought about backup career plans, and these have oscillated from a motorcycle racer to photographer, and now these days I toy with the idea of being a full-time writer, which I never thought I'd say. I remember starting my lab and writing grants. It was like torture to me. I thought, or maybe I used to hate the writing process. I'd procrastinate horribly, and it would just be torture to try and get into that frame of mind for writing. But I think I've learned to love it, or certainly grown to love the writing process. That's probably the majority of my job these days, especially now with the move. I'm just emailing all day. But writing grants, writing papers, this is my happy place now. And I often think about that. Well, what if I, picked up another job tomorrow? And I think I'd be a writer, write some novels. I don't know what genre. That's the only thing. But the idea of just sitting down with my laptop and writing for fun, and also creative writing, seems really appealing to me. So that could be my alternate career plan right now.
Some of the greatest writers, former scientists. So I think that'd be a nice move. And I think I can speak for Arun in saying that our vote for genre would be sci-fi. We're a couple of sci-fi geeks, and I think
would be fun.
Scientists really know how to tell a story. So I'd love to hear yours. Probably be a lot better than my made for Netflix narrative, I'm betting. Finally, what is the best piece of advice that you've ever been given, ever, either professional or not?
This would be in a professional setting, and it comes from George Daly. And this was when I moved to Boston the first time in 2011. I was coming off the back of a postdoc with Magda Januszka-Gertz, and I was going into George's lab, and other postdocs in the lab were telling me, oh, he never takes second postdocs. And so I was like, oh no, I really got to perform, and felt the pressure. And I knew I wanted to continue in an academic career, and I wanted to run my own research lab. And I went into his office in one of our first meetings, and I said to him, George, I need a high impact paper. I need to run my own lab. I need really good papers. How do I do this? And it's embarrassing thinking back to it now. And he gave me the best advice. First of all, he said, calm down. That's not how it's going to work. And it wasn't, I published well in Magda's lab. I'd been publishing a lot of papers, but I felt this intense pressure, especially in the Boston environment. And he said to me, and, this advice he gave to me, still stands today. He said, work hard, be ready, and when the opportunity comes, you'll be able to take it. And at the time, I was like, this is like a riddle. I don't understand what he means by this. And it took me years to understand, really through experience, what that meant. So I left his office. I, kept my head down. I read as many papers as possible, worked hard in the lab. And eventually, through talking to Patrick Cairn, who, he'd started the CellNet project, wrote the CellNet software, and teamed up with Patrick and said, hey, we need to go test these hypotheses in the lab. We need to test how this platform's working. And then that was it. That was the opportunity that turned into the bigger project. And I just think it's fantastic advice. So it's almost like the idea of making your own luck, which I love the idea of that as well. So if you just work hard, you be ready and take that opportunity when it comes, because you're stacking the odds in your favor, that you'll be able to take those opportunities. So that's the best advice I got.
Yeah, that's great advice in life. But particularly in science. And I mean, I'm still relatively, I'm not an old man, I'll say, in science. I've got some years left in me, but I can appreciate that sentiment because, yeah, like it seems like a riddle, but like now it seems obvious, which is you got to keep working and the being ready in particular, right? And just putting yourself around science, staying around science, and when you're ready, or if you're ready, an idea that comes, or an opportunity, or the capability to act, or a collaborator, or a grant opportunity, whatever it is, if you're ready and you can see it for what it is, you can go after it. So that's great advice. Not surprised. George Daly's a pretty smart guy, and I'm sure you're gonna pay that forward, sitting right down the hall from him. That must be very gratifying. Maybe not right down the hall, but metaphorically speaking. Sam, this has been a really, really great conversation, and we appreciate your time, and we wish you the best of luck, although you won't need it, the way you're crushing it. But hope you're going to enjoy this next chapter at the Brigham.
Thank you, and really enjoyed this chat today.
All right, y'all, that brings us to the end of this tweener episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL or by email at info@stemcellpodcast.com with feedback or to suggest guests. This was a off-schedule episode, bringing you more stem cells, more Roundup, more guests, more brilliant insight, and we'll be back at you again in a week with the next episode. Until then, thank you so much for listening.